With section 10.2, we're going to be looking at the activity of the drugs, the antimicrobials, antibiotics, antifungals, the anti-parasitic, whatever. We're going to be looking at how they work, what they target, what metabolic activities they inhibit, what direct damage they could do. Now, keep in mind, when it comes to clinical drugs you're going to be wanting to use in a clinical setting to treat an infection, you're going to be looking at things, you have to keep in mind everything is going to be toxic. There's going to be levels of toxicity. But we can choose to be selective in our toxicity. The idea is to come up with a way to kill, inhibit, do damage to the bacteria, to the fungal, whatever, the parasite, to damage, kill in such a way that you're going to do minimal to no injury to the patient. So what you're going to want to target with not only your development of new drugs or your choice of drugs to use in treatment, you want to go for things that are microbial traits only. Said in a previous lecture in this chapter, cell wall is perfect. Nowhere in your body do you have a cell wall. Pretty much all bacteria do, whether they're gram-positive, gram-negative, they'll have a thick or a thin cell wall, but there's always a cell wall. There's going to be things like if you're coming up with an antiviral, reverse transcriptase for some of the RNA viruses. We don't do that. We can't do that. We don't have the genes or the ability to do that. Perfect target. But you also have to keep in mind that some of the microbes that are going to have to be treated in the clinical setting are eukaryotic, just like us. So the toxicity that you're going to run into that's associated with these drugs is going to be higher than you would see with anything that's antibacterial, antiviral. Because think about it. Antifungal, antiparasitic, well, the parasites, the helminths, the tapeworms and things like that are animal cells. They're pretty much exactly like our cells. So anything you're going to use that's going to damage them, you have to be careful because that's going to interfere and damage the patient. It's just at what concentration are you going to find that it's going to be more damage to the fungal infection, more damaging to the parasites and the parasitic infection, and going to have lower levels of toxicity. When I say toxicity here, think liver damage, possible kidney damage, other damage to other organs. Well, that's going to be minimal, that in the short term may make the patient feel bad, but long term the parasite's gone and the patient will be able to recover. So, mechanisms. Best drugs. Here's going to have one of these 5 mechanisms. First, go after the cell wall. Turns out for bacteria, the cell wall is constantly being renovated. Environments are harsh. Well, especially an environment you is going to be harsh to some of these bacteria. So the gram-positives are constantly shedding that cell wall and then replacing. Now, they're not getting rid of it all at once, poof, and it's gone, but the outer layers are going to be shed. They're going to be stripped off because of damage or whatever. So at the cell, they're going to be making new layers that are going to slowly be replaced underneath by new layers and new layers. So it's kind of like rings. Closer to the cell is the new, further away from the cell, the cytoplasmic membrane, old layers of cell wall. So those older layers get shed off, newer layers made. So you start taking penicillin. Penicillin inhibits part of the cell wall manufacture, part of the cell wall replenishment. So it doesn't kill the cell immediately. What it does is, as those new layers of cell wall are made in the presence of penicillin, they're weakened. The hardier, older cell wall layers over time will be shed, broken off, replaced by new layers that are weakened. They cannot handle the osmotic stress, and given time, the cell will pop. Inhibit. Nucleic acid, structure, function. Turns out, many of these microbes have certain structures within their DNA and their RNA. Their RNA forms certain structures, three-dimensional structures, that ours don't. These three-dimensional structures in the DNA and RNA are signals within the bacterial or the viral or the fungal cell that will trigger certain cellular responses. Well, you inhibit the making of those structures. You do stuff to the structures, have proteins or compounds that bind to those structures, screw stuff up so that those cellular responses within the bacteria, the viral infected cells or the fungal cells, can't happen. You inhibit protein synthesis. Have something that targets ribosomes. This is a great one for bacteria. Prokaryotic ribosomes resemble eukaryotes, but they're smaller, they're less complex. They don't have as many parts as ours do. So you can go through and you can interfere with protein synthesis, gum up the works in the ribosome. Problem is it does bleed over into the patient cells, and there will be some protein synthesis inhibition, minimal toxic levels of that. But it's great. You see a greater effect on the bacterial cell. After a few days, the patient stops taking it, it clears out of the system, eukaryotic ribosomes replenish, go about their business. Can interfere with membrane functions. Don't see this one so much bacterial, because there's so many other great antibiotics, targets, and things. You can see this one with fungal. In my opinion, I've seen, you see this one with fungal parasites. Problem is, fungal or parasites, their membranes are basically almost exactly like ours. They may have some slightly different phospholipids, slightly different sterols and things in their membranes. That this one's going to have high levels of toxicity. Another one, go for folic acid synthesis. I mentioned this one earlier. We don't make folic acid. We do not have the ability. We don't even have the genes to be able to do this. Bacteria do. So you go after folic acid synthesis. You inhibit folic acid synthesis. Many of these microbes are going to run out, deplete their folic acid reserves, and then the cells will metabolically shut down and die. So you look at this figure here from the textbook, points out in a bacterial cell the numerous targets and the drugs that you can go after. Now, if I was a right bastard, I'd make you have to memorize this. And I had under, previous courses said, okay, here's the 20 drugs you have to memorize and what they do, how they work. But it's now a kinder, gentler Dr. Welsh, and I'm not going to do that to you this time.